Good morning. My name is Larry Ryder, and I'm a professor in the Department of Neurology at the University of Tennessee Health Science Center in Memphis, Tennessee, where Elvis was from and rock and roll was born at Sun Studio. And today I'm going to talk to you a little bit about some gene expression studies we've been doing, trying to compare similar patterns in ROHHAD, CCHS, and PWS neurons. So briefly, a history of ROHHAD molecular genetics is somewhat disappointing for a gene jockey like myself. There was a study done in 2011 looking at a handful of genes. Those were found to not be responsible for ROHHAD phenotypes. A larger study doing whole exome sequencing in 35 subjects, which is quite a few for a rare disorder, also found no disease-associated variants. Around that time, a paper came out that seemed to imply that mutations in RAI1 might be associated with ROHHAD, but I've had some discussions with some authors on this paper, and most likely this was a Smith-Magenis case and not a ROHHAD case. And then finally, a few other genes were tested in a larger patient population, but unfortunately, everybody keeps coming up zero in terms of actual gene mutations associated with ROHHAD phenotypes. So to date, no genetic lesions or single gene mutations are associated with ROHHAD. So that puts me in an awkward spot. However, I'm very interested in looking at patterns and patterns of gene expression, even if we don't know individual genes that could be clearly associated with a particular syndrome. So for example, I have some experience with Prader-Willi syndrome, and Prader-Willi syndrome overlaps with ROHHAD in terms of rapid onset obesity and autonomic dysregulation. And then another syndrome that you're familiar with, CCHS, overlaps with ROHHAD, not in terms of rapid onset obesity, but in terms of hypoventilation phenotypes and autonomic dysregulation. But the difference here is both PWS and CCHS have known genes that are associated with these phenotypes, and ROHHAD does not. So my lab uses a unique resource, dental pulp stem cells. These are stem cells that we extract from the teeth of children with a variety of neurogenetic disorders. And we can do that by sending individuals a tube of media, and they put the tooth in the tube when it falls out, and they send it back to us in this box with a consent form and a survey about autistic characteristics. And then we can take that tooth and break it open in the lab and grow these stem cells that on the picture here are these little yellow dots. These stem cells have been made into a variety of different cell types. They're multipotent. They can be neurons, glia, chondrocytes, and adipocytes. We've done several of these things in our lab as well. This is what they look like. On the left side, you can see this flat morphology, kind of look like fibroblasts for the PSCs. And then on the right, you can see these long processes that are more reminiscent of neurons after we differentiate them in using our neuronal differentiation protocol. If you stain these cells, it's a little easier to see the morphology. On the left side, this flat morphology looking like fibroblasts. And on the right side, you see these nice cells with processes and a sort of rounded soma. So they're starting to look like neurons. So we've collected quite a few of these DPSCs in the last 10 years or so. And one reason is it's very easy to collect them remotely because the parents never actually come to Memphis. They send us a genetics report, and we can send them a tooth kit to collect the tooth and return it to us, and then we can make the stem cells. So that's been extremely successful for chromosome 15q syndromes that we work on. duplication 15q, Prader-Willi syndrome, and Angelman syndrome. And so we have a lot of lines in this particular area, although we've also collected lines in other areas. Got a few Potocki-Lupski syndrome, another rare disorder. So we were happy to start this process for ROHHAD and CCHS, and through a collaboration with Dr. Debbie Wiesmeyer, we now have 9 ROHHAD lines and 9 CCHS lines in our collection, as well as our 44 Prader-Willi lines. So these are actual ROHHAD neurons. So these neurons look very much like the control neurons, and we can differentiate CCHS and ROHHAD neurons into these nice neuronal morphologies. I keep calling them neurons. Our lab and other labs have worked on some of these aspects. You can see on the top panel that as the cells mature and are differentiated into neurons, that we get expression of genes like neuroligin 2. There's also some expression of GFAP, indicating that there are glial cells in this culture as well, neurotubulin, which we use as a marker in our lab. And we've actually shown that MAP2 is quite a strong indicator of the differentiation from DPSCs into neurons, and GABA-A is also present in our cultured neurons. So through a grant from the ROHHAD Association and through a collaboration with Dr. Debbie Wiesmeyer at Lurie Children's Hospital, we set out to look at gene expression across ROHHAD, CCHS, and PWS DPSC-derived neurons. The plan was to collect at least 5 individuals and to perform RNA-seq on 4-week-old mature neurons, compare the gene expression both within and across groups, and identify some shared pathways among these syndromes that could be associated with particular disease-associated phenotypes. All the work I'm going to show to you today is the work of a talented graduate student in my lab, Caitlin Victor, who hopefully will be getting her PhD later this year. The general workflow is standard RNA-seq workflow. Unfortunately, we could only collect 3 ROHHAD lines and 2 CCHS lines at the time we did the RNA-seq study. So we did not collect 5 individuals, but we have the DPSC lines now, so we could add to this RNA-seq data. We essentially run a standard profile. We extract RNA, we make a library, we send it off for sequencing, and then we have in-house bioinformatics to analyze the data and find genes that are expressed differentially between control and ROHHAD and across the individual groups of ROHHAD, CCHS, and PWS. So what did we find? So in the ROHHAD specific group, that is transcripts that are expressed differentially between ROHHAD and controls, we found a couple of interesting genes. One of them, FGR, is correlated with obesity. E2F2 is a gene that is associated with neuroendocrine tumors. COL13A1 is associated with respiratory problems. So we found some things that might make sense in terms of ROHHAD phenotypes. Unfortunately, not all of these genes have reagents available for us to check changes in expression at the protein level, which is what we're really interested in seeing. So what I'm going to talk to you about today are a couple that we were able to look at either by Western blot analysis or immunocytochemistry. And those are NETO1, GABA-RA2, and KCNN4. So first we tried our standard protocol of IR Western blots for GABA-RA2, and you can see on the left side that we see a signal in brain. It's a little bit weak, but we see it. And then we see a nice bright signal in our differentiated neurons for control, ROHHAD, and CCHS. Unfortunately, we don't really see a difference among these groups, even though at the transcript level, there is a significant difference between control and ROHHAD. We don't see that at the protein level. For NETO1, we had a different challenge, and that was that we could see nothing by IR Western. So we tried a more traditional ECL Western blot. And in fact, we did see differences between control and CCHS. not so much for ROHHAD, but they are parallel to what we saw for RNA-seq, and that is higher expression and control versus ROHHAD and CCHS. But the gene that really interests us right now is KCNN4, which is a voltage-independent potassium channel. We looked at this gene by immunocytochemistry, and you can see that these little green dots in the top panel for control are really almost gone in the ROHHAD channel and for the ROHHAD subjects. So on the right side, you see beta tubulin. This gives you orientation of where the neurons are. And on the left side, clearly you see this green signal is decreased in ROHHAD. And at the gene expression level, we expected to see a decrease in ROHHAD versus control. So we are now pursuing this particular gene, which is highly expressed in brain and spinal cord and is associated essentially with neuronal function. So another way we analyze our data is by using enrichment analysis. We've done both David and other types of analysis. This analysis is called GORILLA, and it utilizes Go terms and looks for enrichments in particular Go terms in your data set when you plug in a data set of gene expression. So the list of significantly different genes showed 2 genes that are both actually in one particular Go term, and that is CD4 positive alpha beta T cell activation. There's actually only 4 Go terms in that particular category, so this is a pretty strong enrichment. We, of course, also found an enrichment for genes that are involved in the regulation of neurogenesis. There's a lot of these genes, and so it's going to take us quite a while to gather reagents and to do protein analysis for these genes that are associated essentially with the development of neurons that look like they might be involved in ROHHAD. So that brings us back to our table and looking for commonalities and differences among the disorders. So what we did was we took our RNA-seq data that we generated for CCHS and ROHHAD, and we added in some RNA-seq data that we had from Prader-Willi syndrome, and we looked for any transcripts that were common across these different disorders. We actually found quite a few transcripts that are differentially expressed in CCHS and ROHHAD versus control, and those transcripts are all downregulated, almost all of them are downregulated in ROHHAD and CCHS versus control. So on the left side, you see a heat map, and the red indicates upregulation or normal levels, and the blue indicates that there's a downregulation of these transcripts in ROHHAD and CCHS. There were actually 3 transcripts that were shared among all the syndromes, and I'll briefly talk about those in a second. So the green part of the table at the top are transcripts that actually overlap with Prader-Willi, ROHHAD, and CCHS. And one gene in particular that we were interested in is FOXK1. This is a transcriptional regulator that is involved in glucose metabolism and aerobic glycolysis, but also changes during the differentiation process, or should be changing as you differentiate stem cells into neurons. So we've done some preliminary Western blots that I can't really show you yet, but we're hoping that we'll eventually be able to show some differences in FOXK1 across these syndromes. Okay, so today I showed you some gene expression studies we did where we identified significantly differentially expressed genes in ROHHAD neurons that were associated with several phenotypes that seem to make sense in terms of ROHHAD. Those would be FGR for obesity, E2F2 for neuroendocrine tumors, COL13A1 for respiratory problems, DUOX1 for hypothyroidism, but probably most important is this KCNN4 gene that we showed is changing at the protein level between control and ROHHAD, and that's involved in actual neuronal function. So surprisingly, we didn't find much in terms of overlap between PWS and ROHHAD, but we did find 55 transcripts that were changing between CCHS and control and ROHHAD and control, and they were changing in terms of going down. So they were down-regulated in both of those syndromes versus control. And finally, we did see 3 genes that are changing in all 3 syndromes, FOXK1, FBH1, and CNF18. However, at the moment, we don't have Western blot data or immunocytochemistry to prove that at the protein level, these genes are changing as well in these syndromes. So really the next step is to demonstrate that these changes in gene expression are changing as well at the protein level in order for us to figure out how they're associated with various phenotypes. But I think more importantly, maybe we dig deeper into the 55 transcripts that are shared between CCHS and ROHHAD and look for commonalities in terms of pathways that might be good targets for intervention. So again, all the work was done by Caitlin. She's in the middle here, and I've got some other grad students in the lab and some other people, but Caitlin really took this upon herself, and we could not have done any of it without this strong collaboration between our lab and Debbie Wiesmeyer's lab, and the support of the ROHHAD Association. So with that, I'd like to thank my funding agencies that have allowed me to do this work, specifically the ROHHAD Association, but also the Foundation for Prader-Willi Research, who supported a lot of our Prader-Willi work over the years in dental pulp stem cells. And with that, I'd like to take any questions you might have.